Okay, our next speaker will be Dr. Weibke Striegel, and she'll be talking about emerging technology pesticides. Weibke is a biologist whose education and research focused on molecular plant biology. She also came to us as a AAAS fellow. She got her PhD at Colorado State University, where she investigated the biochemical mechanisms underlying plant tolerance to selenium. So with that, I'd like to introduce Weibke.
Thank you, Mike, and good morning. So most of you will be most familiar with conventional pesticides, their registration process, the uses, their applications. And so what I'd like to do in the next few minutes is introduce you to a different type of pesticides, the biopesticides. And here specifically those that have been produced using emerging technologies, which also includes biotechnology. So I'll start by giving you an organizational overview of the Biopesticides and Pollution Prevention Division, and then have 5 examples of pesticide products that are derived from biotechnology and emerging technologies that are either already registered or currently have an experimental use permit. So these are real-life examples. And then lastly, since we're talking about something that is emerging, oftentimes these products pose new challenges for us on the risk management and risk assessment side. So I would like to walk you at the end through one of our examples of one of the products that we recently registered. So biopesticides are derived from natural materials, the animals, bacteria, plants, certain minerals, and they generally have a lower toxicity profile. There are 3 general types of biopesticides. They are the microbial pesticides, which are anything from viruses, bacteria, and fungi, and then they act with a toxic mode of action. They can use the pest itself as a growth substrate, or they could be acting through competitive inhibition. Then there are the biochemical pesticides, and these are naturally occurring substances or synthetically derived substances that are equivalent to naturally occurring substances that have been a long history of exposure to humans and that have a low toxicity profile. And then thirdly, there are the plant-incorporated protectants, and those are evaluated by the Emerging Technologies branch. And you can actually see that the branches so far actually mirror the types of biopesticides that we have within these branches. So I will talk about PIPs a little bit later, actually in the next slide, so I won't go into detail here. But you can see that on these 3 branches are the risk management branches of Biopesticides division. So these 3 branches are also the ones that developers talk to when they first come to BPPD. So these branches also then work with the risk assessment branch, which conducts the scientific assessments of new biopesticide products. And the 5th branch is the environmental stewardship branch, which promotes the safe use of pesticides through non-regulatory means. So this could mean educational outreach through, for instance, webinars. And they have a very successful webinar series on the web that I encourage you to go to. The rest of my talk will focus on the emerging technologies branch and the products that we see in that branch. So these are the 4 general types of products that are evaluated by the emerging technologies branch, and the vast majority are plant-incorporated protectants. Plant-incorporated protectants are pesticidal substances, as Amanda already introduced, that are introduced into the plant. So the plant itself is not the PIP. The pesticidal substances within the plant are the PIP. And so I put up the Code of Federal Regulations definitions because I wanted to make the point that we do not regulate herbicide-tolerant plants. We do regulate the herbicides themselves that are used on these genetically engineered plants in other parts of this program in OPP, but we do not regulate the herbicide-tolerant plants themselves. So the regulations are on the DNA, the RNA, and the protein. And so any other genetic information that has been introduced into the plant that was necessary to create the PIP, we also regulate, and these are inert ingredients. So we regulate active and inert ingredients for PIPs as well. The second group here is the genetically engineered microbials. So those are all non-naturally occurring microbials that are then otherwise would have been regulated by the microbial pesticides branch. We have products that are microbials introduced into mosquitoes for mosquito population suppression. And we also evaluate genetically engineered mosquitoes, and that is relatively new This is since October 2017, where FDA clarified that these types of products are not new animal drugs if they are used for mosquito population control. Now I would like to go over some of the real-life examples. And here we have actually 5 examples. 2 are plant-incorporated protectants they're active against corn rootworm. One is a genetically engineered plum against plumpox virus. We have a genetically engineered virus that combats a bacterium that causes citrus greening. And lastly, the Wolbachia bacterium that was integrated into the Asian tiger mosquito for mosquito population suppression. So corn rootworm is of significant economic importance. The management is relatively expensive. It can upward of $1 billion per year. And so the larvae feed on the roots, and then the corn plants lodge and cannot be used. So I will not go too much into detail about corn rootworm here because my colleague Alan Reynolds will use corn rootworm as a case study later on this morning. So corn rootworm is usually managed through conventional pesticides in conjunction with crop rotation, for instance, with soybean. But again, there is a heavy economic burden on this, so any type of different management tools are appreciated. So one of the biotechnology approaches here was to use a protein that was first identified in the soil bacterium Bacillus thuringiensis and has then been introduced into the plant so that the plant is now able itself to produce this type of protein. Now, Bt proteins, these toxins have been used over decades in agriculture as exogenously applied to the plant, and they're currently also used in organic agriculture. So what happens when the larva of the corn rootworm eats corn pip now is that it ingests these proteins, and the proteins get processed within the digestive tract of the corn rootworm and then are able to bind to receptors inside the gut of the corn rootworm, which then leads to perforation of the gut membrane. And so that leads to reduced nutrient uptake and invasion of microbes and then the demise of the corn rootworm. So thus far, BPPD has seen more than 100 PIP products, not all of which work on rootworm. There are many Bt proteins, like I said, over 100 products of these. So what happens is that we want to obviously prolong the lifetime of these pesticides and mitigate resistance in the long run. So what you can do is you can not only express one protein in a corn plant, but you can produce many proteins. And if you produce proteins that target a single pest, then it's called pyramiding the protein or these traits into one corn plant. So one of the newest traits that was integrated into corn is not a protein, but what Amanda also introduced, an RNA. So the gene now produces an RNA molecule that then targets a gene within the corn rootworm. So the gene is called DV-SNF7, and this is an essential gene for corn rootworm. So if you remove that gene, the corn rootworm cannot fully develop and dies. Corn rootworm is able to take up the RNAi throughout the body. This is not specific, but relatively unique to corn rootworm, so it doesn't happen with all organisms upon ingestion. So here, double-stranded RNA from the corn plant is ingested, and then the essential gene is downregulated in the corn rootworm. Now we're going from a macroorganism to a microorganism, and specifically here, the plumpox virus, which causes Charcot disease. Upon infection, as with many viruses, the plant is basically hijacked to produce more of the virus itself, which includes RNA and the coat proteins that make up the virus. So then the viruses spread throughout the plant and then are vectored by aphids throughout the orchards. So it's really hard to manage this, even with insecticides, because you need only a few aphids to transmit the disease. And so one of the things that has been done in the past is to basically burn down entire orchards, which you might imagine is not very sustainable in the long run. So one of the emerging technology solutions here was to engineer a plant that you can imagine it to be immunized, if you want that. It's not completely analogous, but you can think of it that way, to this particular plumpox virus. So this entire tree has been engineered to produce a double-stranded RNA again. In this case, it matches to the RNA of the virus itself, and then this double-stranded RNA is recognized by the plant as aberrant and degraded. You still have the affected plant, but the spread of the virus is significantly reduced. We have registered this product, which has been developed by USDA in 2011. Well, as you may imagine, creating a wholly transgenic tree can take its time because you have to grow up the tree. So sometimes it is more beneficial, or it's a different approach to use a virus to combat the disease. And one of those examples is citrus greening, which has been devastating the U.S. citrus industry for the last decades. Citrus greening is caused by a bacterium, Liberibacter asiaticus, and which has actually not been cultured yet, which makes it even harder to find something that is active against this bacterium. The bacterium infects the tree in the phloem, which is kind of like the sugar highway, so you get all these very low sugar oranges. So these cannot really be sold. The bacterium is vectored by the Asian citrus psyllid, which is not native to the U.S., and so together they kind of create this problem in Florida especially. And this is one of the many statistics that is out there, which is a 2/3 decline of citrus production over the last 20 years. So again, how do you manage it? You can spray for the citrus psyllid, but since you have orchards and these trees are many decades old in some cases, the spread is really hard to contain. And so one of the ways to deal with it again is tree removal. Or you can fortify the trees by enhancing their nutrition as well. So here the approach was to use a virus to combat a bacterium. So the virus that was used is citrus tristeza. It is a ubiquitous virus that causes relatively mild symptoms in the trees. The virus itself was engineered to contain a protein that was originally identified in spinach. So there are a lot of different parts to this. There has been exposure to these spinach proteins through normal ingestion, and actually in spinach it helps the spinach plant combat bacterial infection. So now if you take an infected citrus tree that is infected with a bacterium, and then you co-infect it with this virus containing the spinach defensin protein, then the plant is hijacked to create this spinach protein, which then combats the bacteria. A very neat way with multiple players to get to what you want, which is the healthy citrus fruit. We have issued an experimental use permit for this particular product, and there's also a similar product under an experimental use permit which uses the same types of techniques. However, the spinach defensin proteins were genetically engineered into the plant, creating a plant-incorporated protectant. So now we are switching gears a bit to the control or the suppression of mosquitoes. So the best way to get rid of your mosquitoes in the backyard is usually to reduce the breeding grounds. But if you look at the picture on the left, it's really easy to imagine that there are all these nooks and crannies that one cannot really get to or can forget about. So you can use aerial sprays to generally get rid of the majority of the mosquitoes in an area, or you can use larvicides in conjunction with that. But there will always be a few that will make it through, as kind of illustrated here on the right. They just are under leaves or under tables or whatever it may be. So last year, we registered a product that takes a somewhat different approach. And this approach is to introduce a naturally occurring bacterium into male Asian tiger mosquitoes, and then release those mosquitoes into the environment. So you first have more mosquitoes to have less mosquitoes. The main points here are that the releases are only male mosquitoes, so male mosquitoes do not bite humans. And secondly, it is species specific because it is dependent on the mating of these male mosquitoes with the female mosquitoes that are already in the environment. Wolbachia is present in over 60%, estimated over 60% of insect species and in some crustaceans. And the way it works is that on the top half there, you would have two wild mosquitoes, the males and the females, and they would contain this same strain of the Wolbachia bacterium. Now, when they mate, they create viable offspring. The offspring will then also have the same strain of Wolbachia. Now, in this new case, a different strain of Wolbachia was introduced into the male mosquito. Those are released and then mate with the females that are present in the environment, and then the eggs will be laid, but those eggs will not survive. They cannot develop into adult mosquitoes. And that is through a mechanism called cytoplasmic incompatibility, which basically means that the offspring doesn't get all the genetic information necessary for their survival. So as you may imagine, releasing a live mosquito into the environment is a little bit different than putting a larvicide into a certain body of water. So we had to think about certain ways on labeling and all these other things. So the way you would use this particular product, and you can see it here on the right, they're coming in this particular case in little cylindrical cartons. You go to particular spots in the treatment area and you open them up, and then they release and they fly out, and then they seek out the females, which is kind of like they know where they're hiding, where maybe your aerial sprays won't get into. One of the advantages of this is, too, you can actually use this in conjunction with an IPM program, integrative pest management program. Larvicides you can use either way. With adulticides, if you spray, you can use it too, you just have to time it right. So it's actually an additional thing you can use in your IPM program. Some other considerations that we had with this particular product was the dissemination of the mosquitoes. How far do they fly? At what intervals do we have to release these mosquitoes in a certain treatment area? The health of the mosquitoes, this is nothing you can store for a month and then release. So there has to be some sort of timing and viability. How many males do you have to release? Because remember, there are also males out there that basically now have to be overwhelmed by all these other males that come in that then preferentially mate with the females that are in that area. So there has to be some monitoring up front. And then, of course, accidental female releases. Males do not bite, but females do. So what is the percentage of females that may be released? And how do we make sure that they maybe do not carry any diseases? So these are just some of the considerations that we had and overcame before we issued the registration last year for the Wolbachia bacterium. And here I wanted to point out that we're regulating the Wolbachia bacterium as a microbial pesticide. And with that, I thank you, and I take any questions that you may have.